Hi everyone, my name is Grant. This is my CRISPR video presentation. CRISPR-Cas9, or clustered regularly interspace short palindromic repeats, basically is a group of DNA sequences found in genomes of bacteria and archaea. It is used to edit and rewrite DNA to create desired outcomes in everything from food to animals. This really incredible technology allows us to alter DNA through gene editing and to eliminate undesirable traits of DNA from any species, and that including us, humans. The process of CRISPR comes from altering genes by using precision to cut DNA sequences and letting natural DNA repair processes take over. Now, the method of cutting DNA consists of a Cas9 enzyme and the guide RNA, which is how the DNA sequences link up. Now, the first initial CRISPR type technology discovery was by accident. When sequence of DNA cloned from E. coli genomes was kind of discovered by Yoshizumi Ishino back in 1987. While Ishino did not really understand the repeated sequences or the function of the interpreted cluster repeats, he and his team honestly would go on and be the first ones to kind of discover and really create the idea that the application of biomedical technology and research surrounding CRISPR technology would be something for the future. He really didn't realize that at that point. Now, Francisco Mexico in Spain back in 1992 was the first to introduce the possibility of gene editing and utilizing it, really. He discovered the different species of bacterium had an immunity response to virus infection by altering their DNA to a similar sequence to the virus. That's inserting that sequence into their own genome, which is amazing. Now, Emmanuel Charpentier and Jennifer Doudna won the Nobel Prize in Chemistry back in 2020 for actually developing and adhering CRISPR technology to the human genome. A quote by Doudna would be, clinical trials result and demonstrate that CRISPR is safe. It's an effective way for treating and curing human disease. It's an extraordinary advance in technology, and at that time it was only 10 years old. Doudna also Doudna and Emmanuel were both the first women to actually win the Nobel Prize in Chemistry, and the first team of women actually. The discovery of the CRISPR-Cas9 bacterial immune system use showed the world that it can be used as a gene editing tool. They actually didn't get credit for that originally because they had originally discovered it and published it in the Science journal back in 2012. By the time it was 2020, they got the Nobel Prize. A lot of scientists around the world had actually adapted and started using CRISPR technology for their own different purposes. Touching on those purposes, the cell and gene therapy and agriculture uses is what I'm going to touch on in this video presentation today. Now, CRISPR technology and gene and cell therapy is used to study various genetic diseases that happen in humans, such as muscular dystrophy, cystic fibrosis, hearing loss, sickle cell disease, and hemophilia, also cancer. In some of these fields and some of these diseases, the alteration to people's DNA using CRISPR technology has been successful, and people who suffer sickle cell disease and various other ones that I mentioned no longer live in that pain and stress. It's still being tested, but it's also been implemented for cancer. They create and basically edit types of T cells so they can transplant it back into the patient after they use CRISPR technology, and the patient's own T cells that are normally not active during cancer actually are attacking the cancer cells. It's amazing. Now, in agriculture, CRISPR technology honestly is doing something that's not necessarily new to being done in agriculture. Farmers have always bred crops and animals to bring out different desired traits, specifically for consumption and just to make them more abundant. Now, CRISPR tools have been used to modify plants' DNA and make those wanted changes to breed those traits that would normally take a lot of time, resources, and just extensive and expensive methods. CRISPR, for example, was able to create low gluten wheat crops and disease-resistant rice, which makes so much difference in our future as the world kind of becomes more dangerous to crops and it becomes more difficult to grow and keep crops. Now, if I were to have a laboratory and I was able to use CRISPR, I would implement it to study effective ways of treating genetically related human mental illness. Different illnesses such as schizophrenia and bipolar can benefit significantly from using CRISPR technology as a treatment method to hopefully alleviate the genetic mutations that are linked to these diseases and various other mental illnesses. I know that it's possible, hopefully in the future, to help people with ADHD, which is something that I personally suffer from. Now, in moral and ethical issues, there are some standing in there for CRISPR technology. It all surrounds gene editing technology in general. While it is an incredible advancement, specifically things like mosaicism, which is a condition where one possesses two or more different cells with other genetic lines due to a genetic mutation. Now, it's not something that seems to happen as often, but CRISPR technology could possibly mess something up in someone's DNA sequence and create another genetic mutation or just create something different that is not meant to be in our DNA. Now, another ethical possibility, basically, we don't know how well and how expensive CRISPR technology is going to be used and distributed in the future, but it might only be for people who can actually afford to receive this gene editing technology, which is not fair because it could save so many lives and be so impactful on people who are suffering from the various diseases that I've mentioned in this video. Also, it could create divided issues for people who have it available to it and people who can't. Another ethical issue could be discrimination against people who have received gene editing technology. Basically, insurance companies who see that someone has used gene editing technology might not want to insure or cover them for various other illnesses or different medical issues in the future, and that could be detrimental on people who maybe, while they had gene editing for some type of genetic disease, maybe they hurt themselves and can't get the insurance companies to cover them because of their gene editing. Lastly, the idea of clinical trials going differently than planned and having lasting genetic mutation effects on those tested with CRISPR-based therapy could be a risk in actual human clinical testing. I believe that people need to be tested on, but at the same time, it's scary to think that something could happen to someone where their genes were altered when they were hoping to have something cured. Overall, CRISPR is amazing technology, and I appreciate having the opportunity to talk about it and share this with you. Thank you so much, guys. Have a nice day.